You've seen a lab report, right? It's a piece of paper with your name on it and, well, a whole bunch of numbers. But behind every single one of those numbers is this entire hidden world of precision. It's a silent language that turns a simple drop of blood into a really detailed story about your health. Today, we're going to pull back that curtain and look at the math that makes it all work.
I mean, seriously, just think about it for a second. How could a machine take this tiny, tiny sample and spit out these incredibly specific numbers about everything from your cholesterol to your blood cell count? It kind of feels like magic, doesn't it? Well, it's not.
And this right here is where our trust in modern medicine really comes from. It's not built on guesswork. It's built on a super strict universal language of numbers. This ensures that a result from a lab in, say, Tokyo means the exact same thing as a result from a lab in Toronto. And it all kicks off with some really fundamental rules. So let's start with the basics, the grammar of this language, if you will. Before we can even get to the big, fancy calculations, we all have to agree on how to write the numbers down in the first place. And this is all about being honest and accurate with our measurements. You might remember these as sig figs from high school chemistry, and they're not just about rounding numbers for the sake of it. Think of them as an ethical promise from the lab to you. They stop a result from looking more precise than the test can actually be. It's all about reporting only the digits that we can truly stand behind. And here are the rules that keep everything honest. The best way to think about it is like a chain. Your final result is only as strong as your weakest link. So if you measure one thing with a super-duper precise instrument, but another thing with a less precise one, you have to report your final answer based on that less precise measurement. It's all about making sure that final number is a true, honest reflection of the work that was actually done. Now, to make sure everyone around the globe is speaking the same mathematical language, labs use what's called the International System of Units, or SI units. It guarantees consistency. As you can see here, a base unit like the kilogram for mass gets tweaked for lab use into something more manageable, like grams. And of course, being part of a global system means you've got to be able to convert between different units. While most of the scientific world is on board with Celsius, places like the US still commonly use Fahrenheit. So these simple formulas are absolutely crucial for making sure temperature-sensitive tests can be understood and repeated anywhere in the world. Okay, so we've got our rules for how to write the numbers and what units to use. Now let's kind of get our hands dirty and look at the math that goes into actually prepping the samples for testing. Diluting things is one of the most common things that happens in a lab. A lot of the time, a patient's sample, maybe your sample, is just too concentrated for an instrument to read it accurately. So what's the solution? You dilute it. You bring the concentration down in a very, very precise and mathematical way. Let's just picture how this works with something called a serial dilution. We start by taking one part of our original sample and adding it to one part of a diluent, something like saline. Just like that, we have a 1-to-2 dilution. The concentration is now exactly half of what we started with. Now, if that's still too concentrated, we don't go back to the original sample. Instead, we take one part from our new 1-to-2 mixture and add it to another part of the diluent. See what we did? We just halved it again, creating a 1-to-4 dilution from where we first started. And we can just keep on going. We take a bit from that 1-to-4 tube, repeat the process, and boom, a 1-to-8 dilution. This whole serial dilution method is a super quick and accurate way to get a whole range of different concentrations to test. And this simple, beautiful little formula right here, this is the golden rule for all dilutions. It basically says that the volume you start with times the concentration you start with is always equal to the final volume times the final concentration. What that really means is, while the solution gets more spread out, the total amount of the stuff you're trying to measure never actually changes. So we have our rules, our units, and our dilutions. Now for the fun part. Let's see how they all come together to create some of those key numbers you might actually see on your own lab report. First up, let's grab an example from hematology. That's the study of blood. The mean corpuscular volume, or MCV, is a really critical piece of information. In simple terms, it tells your doctor the average size of your red blood cells. And it's calculated using this pretty straightforward formula. We're about to decode what HCT and RBC are, but just notice for now how it's really just a matter of multiplying and dividing a couple of key measurements to get this powerful answer. Okay, so here's the decoder ring. The hematocrit, or HCT, is just the percentage of your blood that's made up of red cells, and the RBC is simply your total red blood cell count. By putting these two values together, we can figure out the average volume of a single red blood cell, which is a huge help when it comes to diagnosing things like different types of anemia.
All right, let's hop over to another department, clinical chemistry. The anion gap is a really clever, indirect calculation that helps doctors check out a patient's acid-base balance, which is absolutely vital for, well, your whole metabolism. The formula works by looking at the balance between positively charged ions, in this case sodium, and the main negatively charged ions, which are chloride and bicarbonate. By subtracting the big negatives from the big positive, doctors can find this gap that can point towards certain metabolic issues. It's an incredibly powerful tool, and it comes from just simple subtraction. Okay, so we've seen how the numbers get calculated, but that brings us to maybe the most important question of all. How do we actually know the numbers are right? I mean, that's everything, isn't it? This final piece of the puzzle is all about statistics and quality control. And the key tool for that is something called standard deviation. Put simply, it's a measure of consistency. If a lab runs the exact same sample 20 times, a tiny standard deviation means they got almost the exact same result every single time. That is the sign of a very precise, very reliable test. And this chart shows you exactly how labs use this idea every single day. They run these special control samples where they already know the right answer, right alongside all the patient samples. The results for those controls have to fall within a specific range, usually plus or minus 2 standard deviations of the known value. If a control ever falls outside that range, like that bar on the far right, the entire batch of results gets stopped, flagged, and retested. You see, this whole mathematical framework, from the sig figs at the very beginning all the way to these quality control stats at the end, it's a massive system of checks and balances. It's all designed to guarantee that the numbers on your report aren't just numbers. They are a trustworthy reflection of what's going on with your health. Because now you know it's not some kind of magic trick. It's a story, and it's written in the universal language of math. It's a story about precision, about accuracy, and about trust, all designed to give you and your doctor the clearest picture possible. So, what will you see in those numbers next time?